time is sort of a small miracle. Okay. If we delve into this, right, this is what we're going to cover today. All right. Note that you've got basically good microbes living in every body cavity that's exposed to the outside world. Okay. And so that includes the surface of your skin. That includes the mucous membranes that line your GI and your respiratory and your urinary and your reproductive. Okay. It includes even ear canals and your orbits. Okay. All of that is populated with microbes, most of which are beneficial or at the very least not harmful to you. And because of that, they take away areas where pathogens could colonize and grow. All right. And that's a layer of protection that's in addition to what your body already has to defend you against disease, which we're going to go through each of those today. Okay. Um, Relationships between organisms follow a variety of possible scenarios. Okay. When it's a useful relationship, we call it a symbiosis. All right. Now, sometimes a symbiosis is useful to just one or other member, and sometimes a symbiosis is beneficial to both members. All right. It all depends on the type of relationship they have. In mutualism, both members are going to benefit. All right. In commensalism, one organism benefits and the other isn't harmed or helped. Okay. So depending on the outcome, we can classify it as mutualism, commensalism, parasitism, or amensalism, right? An example of mutualism would be the gut bacteria that live in your colon. All right. They break down the waste products from the food that you eat, your digestive system, and in turn, they give you B vitamins and vitamin K. And so they get a place to live and a constant food supply, and we get their byproducts as a result. They also help to break down our waste products. So that's beneficial for both. Okay. In parasitism, one organism benefits and the other is harmed. Okay. Um, Example, right? Species of bacteria, protozoa, and algae and fungi can be microscopic human pathogens. Larger pathogens include parasitic worms and biting arthropods. Okay. An example of parasitism would be a tick on a dog, right? The tick is the parasite and gets the benefit of the blood meal, and the dog is harmed because he's getting an injury, and also that could be an avenue for infection. Okay. In amensalism, one organism can hamper or prevent the growth of another without being affected by the other organism. A familiar example is penicillium, which is a mold that produces an antibiotic called penicillin, which is a chemical capable of killing a wide range of bacteria. Okay. So it inhibits a limited area, but of course it doesn't wipe out the entire bacterial population. Normal flora, which is your microbiome, happens essentially even before you're born. Okay. You get colonized in utero, in fact. And we know this because we've examined the stools of newborns and have found microbes in it. All right. They come into contact through vaginal birth in the birth canal. When they breathe, they inhale microbes. When they feed, they ingest them. Throughout life, the microbes establish residency in all the mucous membranes and cutaneous membranes. Okay. The resident flora are going to remain part of you for the rest of your life, while transient flora are found in the same location as residents, but only remain for a short period of time. Okay. An example of resident flora is Staph epidermis, which lives on the surface of your skin, or E. coli, which lives in your large intestine. Transient flora can't survive for reasons such as competition with your good microbes for nutrients, for areas of colonization, as a result of defense by the immune system, and chemical and physical changes in you. Okay. An example would be Bacillus laterosporus, sometimes found in the intestine. When present, the organism helps keep fungal populations such as Candida in check. Okay. So it's a battle, right? You can think of your body as a hotel, and um, most of the guests that live in a hotel body are good, right? Or at least not harmful. And then only a few of them are going to cause trouble. Now, opportunistic pathogens are those that take advantage of a weakened immune system. Okay. Um, they're normally around in small numbers, but when the immune system isn't functioning the way that it's supposed to, they take off. Okay. A perfect example would be thrush. Okay. I don't know if you've ever seen anybody with thrush, but when they open their mouths and say, it's covered with white fuzz, and that white fuzz is an organism called Candida albicans, which is a fungus that's always around, all right, but normally is kept in check by the immune system. Okay, so example, thrush. You would see thrush, for instance, in the oral cavity of somebody with AIDS, or in the oral cavity of somebody undergoing chemo or radiation. Would be an example of where you pick that up. Okay. Normally, they don't cause trouble, right? The balance between normal flora and host is maintained, but when we interrupt that, microbes of normal flora can become pathogenic. Organisms don't cause disease in their normal habitat in a healthy person. Okay. This is one of the reasons that your immune system is so critically important, all your defenses. Okay. So a compromised immune system is the doorway for these guys to take off, right? Changes in normal flora, which are normally protective, can result in the pathogen overgrowing. The entrance of the normal flora into areas of the body where it isn't present under normal conditions can cause a problem. Any factor that's going to suppress or weaken immunity can allow this pathogen to cause infection and disease. Examples, right? Acute and chronic diseases, especially involving immune suppression, right? Malnutrition, stress, age, the use of radiation and chemo in the treatment of cancer, and the use of immunosuppressive drugs in transplant patients. All of those are doorways for opportunistic infection to take off. When the normal flora changes, it can allow one of the members to become an opportunistic pathogen and thrive. Examples include vaginal yeast infections, Candida albicans, right? Yeast infection. You probably heard that before, and we gave the example of thrush earlier. Okay. Entrance of normal flora where not present under normal conditions can occur after injury in a burn victim, or even when an intestinal organism such as E. coli enters the urethra, where it can produce a UTI. Right? And that is something that's more common in women than in men because of the short urethra and the proximity to the anus. Right. So guys can get them, but it's usually only seen in men that have a condition called prostatitis, where the prostate gland enlarges to the point where you can't pee. Right. And what will happen then is that the bacteria that are on the surface of the skin can work their way up the urethra and generate a UTI. Okay. Hold on a second. Let me take care of this. Yeah. Okay. All right. So opportunistic pathogens, right? They're a risk. Stages of infection, right? Contamination is just the presence of microbes on or in the body. Okay. Contaminants reach the body in food or drink, by air, or by fomite, or they can be introduced by wounds, insect bites, or sexual intercourse, right? In infection, the microbe has to gain entry into the host and its tissue by the appropriate portal, meaning, an access point. An infection doesn't necessarily produce disease, right? The outcome of contamination is going to vary. Some microbes remain at the site where they first came into contact with you, such as those on the skin and the mucous membranes, where they don't cause harm and become part of the resident and transient flora, okay? And are kept in check by your good microbes, right? What are the portals of entry? Well, there's a bunch of them, okay? These are sites where pathogens can get in. Source of infection can be exogenous, meaning coming from outside the body, or endogenous, meaning coming from inside the body. Okay. portals generally the same areas that support normal flora, your good guests in the hotel body. Most pathogens have their preferred portal of entry. If a pathogen enters the wrong portal, the infection won't occur. And some infections agents can enter by more than one portal, right? So portals, right? Skin, mucous membranes, the placenta, the parenteral route, meaning through an opening in the skin, an injury, okay, or through surgery, right? Strep and staph have adapted to a lot of portals of entry, such as the skin, the urogenital tract, and the respiratory tract. So you can see here, right, some of the different portals of entry, okay? An example of a portal of entry that is exclusive to women is the birth canal, right? A woman's reproductive system is open to the outside world and to the abdominal pelvic cavity. And because of that, there's a mechanism for pathogens to move from the outside all the way into the abdominal pelvic cavity. And that can produce a condition called PID, pelvic inflammatory disease. Okay. Men don't have that problem because the male reproductive system is only open at one end. Okay. The testes are continuous with the seminiferous tubules, which are continuous with the urethra, right? But in women, the vaginal opening leads to the uterus, leads to the fallopian tube, but the fallopian tube is open to the abdominal pelvic cavity in the female. It's not sealed to the ovary. And so that's how it can get inside, right? Your skin is one of the ways that a pathogen can get inside, right? It's a thick layer of keratinized dead cells. Pathogen get in through natural openings like hair follicles and sweat glands. And they can get in through injury, right? Laceration, puncture wound, burn, okay? All of those are ways to get inside. The outermost layer of the skin is one of your first lines of defense against invasion. The larvae of some parasitic worms and some arthropods burrow through the skin to get underlying tissue access, and some fungi use their digestive enzymes to get entry into soft tissues. These are called virulence factors, weapons that these pathogens can use to produce disease. Right? Normally, right, your skin keeps you safe because your skin is made up of epithelial tissue on the surface, which is constantly made and shed. Okay. And that cuts down on colonization by pathogens. In addition, you produce sweat and oils, and that flushes the surface of the skin and washes a lot of the bad stuff away. And also, the good microbes that are there metabolize the oils and turn those into acids. And that, along with the high salt content of your sweat, produces something called the acid mantle. Okay. And the acid mantle is one of those things that does not promote the growth of a lot of pathogenic microorganisms. A lot of bad actors don't dig low pH and high salt. That's just not their thing. Okay. What else? Well, just beneath the epithelial layer is connective tissue, and in the connective tissue are white blood cells. And the white blood cells are basically waiting for anything that leaks through so that they can eat it, chop it into little pieces, and then present it on the surface of something like a dendritic cell or a macrophage so that your adaptive immunity can be aware that there's an organism that's trying to gain access and cause trouble. And what happens then is that the lymphocytes that recognize those little particles are stimulated to divide and make a little army that goes out and kills whatever that antigen is attached to. So it's a pretty good system, okay? The last thing we want, the worst possible outcome, is a blood infection, and that's called septicemia. And those are microbes that are growing and thriving in the blood. And if that happens, it can spread to every organ in the body and it can be lethal. Okay. This is often what happens when you get, a traumatic event, like say a ruptured appendix. Okay. That makes a hole between the GI and the abdominal pelvic cavity, and the feces is packed with microbes, and that's going to inflame the peritoneum, lead to peritonitis, and then from there it'll get into the blood, and that's septicemia. And if we don't act quickly, that'll kill you. I actually know somebody who died that way. Mucous membranes are going to line the body systems that are open to the outside world. So this is the GI, the respiratory, the reproductive, the urinary, okay? And the thing about a mucous membrane is that it makes mucus, right? And mucus is a sticky liquid made by things like goblet cells that traps pathogens and particulates and flushes them out of the tract so that they don't get a chance really to get deeper inside. But a mucous membrane also, in addition to having an epithelial lining that reproduces rapidly and gets shed and made again over and over and over, it also has connective tissue beneath it, and in the connective tissue are huge teams of white blood cells waiting for trouble, right? So an example, right, the GI tract, a port of entry for pathogens present in anything that you eat or drink, right? Enteric bacteria include Salmonella, Shigella, Vibrio, and strains of E. coli that can cause disease. Viruses include poliovirus, Hep A, echovirus, rotavirus, protozoans, Entamoeba histolytica, Giardia lamblia, and helminths such as trematodes, cestodes, and nematodes, right? Example would be something like the tapeworm. Microbes that are going to survive in the GI have adapted to the action of digestive enzymes and the differences in pH. Right? In the respiratory tract, right, it's the most frequently used port of entry because the air you breathe is not sterile. Okay, pathogens enter through air, through dust particles, moisture, and respiratory droplets from affected people. Bacteria are causative agents of sore throat, meningitis, diphtheria, and whooping cough, while viruses can cause the common cold, flu, measles, mumps, rubella, chickenpox. Right? The mucous membranes that line the upper respiratory tract are continuous with the membranes of the sinuses and the auditory tubes, and so pathogens can be transmitted from any of these locations to another. That's why a lot of times doctors will specialize in eye, ear, nose, and throat, right? Because these are connected regions. Okay, the ear, the nose, the throat are all, and the sinuses are all one big chamber, basically. And so infection can move from one to the other relatively easily, right? In the respiratory tract, the upper respiratory is lined with something called the mucociliary escalator. Okay, let's write that down. And what that is pseudostratified ciliated columnar epithelium that produces mucus, and then the cilia move the particle and pathogen laden mucus up into the oral cavity. And there you can either swallow it or spit it out. Right? The same type of material lines the nasal cavity and moves that material into the oral cavity. That's why sometimes when you have a cold, you get something called postnasal drip, and that can irritate your throat. Okay. If you swallow it, the stomach acid will take care of most of the pathogens that are in whatever the mucociliary escalator moved up to your oral cavity. And of course, you spit it out, it's gone. Okay. So again, the benefits of having the appropriate material line the appropriate cavity. Okay. Now, if we look at your genital tract, right, pathogens are usually contracted by sexual contact. Women who are not sexually active are susceptible to lower UTI because of the close proximity of the anus to the urethra. Opportunistic infections by E. coli can occur, as well as vaginal yeast infections produced by Candida. Okay, vaginal yeast are common in women who are under stress, who are taking birth control pills, antibiotics, or steroids, and who are pregnant, or because of other factors. Okay, and of course, in the lower right there, you're looking at the lining of the mucous membrane of the urogenital. Okay, So one of the things to appreciate with these yeast infections is that it's a battle between the bacteria and the yeast to keep a healthy surface, right? That's one of the reasons sometimes if you take antibiotics and you wipe out your lactobacillus that lives in the urogenital, the yeast can overgrow and cause a yeast infection. Your conjunctiva is the inner lining of the eyelid that runs down to the area where the sclera of the eye, which is the white of the eye, meets the cornea, and that's a region known as the limbus. Right? It's highly vascularized, and it's a barrier against infectious agent. Some bacteria can attach to this membrane and inflame it, and that's something commonly called pink eye, but the clinical term is conjunctivitis. Right? Examples of bacteria that can attach include Haemophilus aegyptus, which could cause pink eye, Chlamydia trachomatis, and Escherichia gonorrhoeae. The placenta is the attachment point between the umbilical cord and the uterus, and its purpose is to provide an exchange point for nutrients and waste products between fetus and mother, as well as oxygen and CO2. Okay. But it's also a barrier to infection, and it also allows maternal antibodies to cross into baby's blood to provide something called natural passive immunity. Okay? It's formed from both maternal and fetal tissue, and it's one of the ways that we protect the mother from the blood of the baby. Right, the mother blood and the baby's blood never mix except at birth, when the placenta is torn from the uterus. And during development, the placenta brings the baby's blood into close contact with the mother's blood, but they never mix. And then we do exchange through a combination of both active and passive transport. And this is one of the reasons why, especially during the first trimester, we warn mothers not to use alcohol, not to take recreational drugs, and to stay away from people that are sick, because that's the time when the baby's organ systems are being developed from the ground up. And if we make a mistake during that period of time, it can be disastrous. It could result in birth defects or even a spontaneous abortion. Okay, so that placenta is one of those things that, it blocks out most stuff that's harmful to the baby, but some stuff can penetrate. Okay. Parenterally, okay, the enteral route means you ingest it. The parenteral route means that it gets into your body some other way, through an injection, through an injury, okay, through a patch. All right, it's technically not a portal of entry. Pathogens are directly introduced into subcutaneous tissue through wounds, okay, cuts, bites, stab wounds, deep abrasions, and surgery can all allow stuff to get inside. Now, that said, all right, we should touch on something that's important, which is that non-polar compounds can go right through this, okay? And by non-polar, we mean lipid soluble, all right? They can penetrate the skin because the plasma membrane is not a barrier to them. They go right through it. Examples would be things like steroids and toxins like tetraethyl lead, and certain types of drugs. Nicotine can penetrate through the skin. Okay. So here we get into a little bit of history, which I think is kind of interesting. Back in the 1800s, one of the most miserable jobs was that of chimney sweep. And what you had to do there is you would lower a person down into the chimney to remove the coal dust so that the smoke wouldn't accumulate inside the building and kill everybody from smoke inhalation. But it turned out that the coal dust had a compound in it called diethylstilbestrone, which is a lipid soluble carcinogen. Okay. And so what happened to a lot of these chimney sweeps is that they ended up dying of testicular cancer. All right. because it just seeped in through the skin and got into the deeper tissues and made cancer cells. Another common occupation at the time was haberdasher. And those are the people that sold hats. Hats were a big thing back in the 1800s. And for reasons that are unclear to me, the hat bands were treated with elemental mercury, all right, which is a liquid metal, which is nonpolar and can seep right through the skin. And what it does is it forms a neurotoxin inside the body, and it compromises brain function, and you end up with cognitive deficit. And so a lot of these haberdashers would get high doses of elemental mercury and would lose cognitive function. And this is actually the true basis for the character in Alice in Wonderland called the Mad Hatter. Okay. If we flash forward to the 60s, okay, lead was an additive in both paint and gasoline. All right. Tetraethyl lead used to stabilize gas mixtures, all right, so that they performed as a better fuel. The problem, of course, is that the tetraethyl lead, is a neurotoxin. And so it would get into the environment and would seep into the ecosystem and would damage the wildlife and would damage anything that consumed the wildlife that had these high doses of lead in it, such as fish. Okay. Another example was the lead paint that used to be used, right? The children would eat the lead paint chips and they had a sweet taste to them. And what that would do is produce cognitive deficit, and they would perform poorly in school. So we took the lead out of the paint and the gasoline to avoid that. Another very popular activity at the time was the use of a fungal derivative called LSD. You may have heard of it. It's a hallucinogen. Okay. It was promoted at the time by Dr. Timothy Leary as a mind expanding drug, but actually all it did was damage the brain. And people would use LSD by taking it on sugar cubes or by touching it to their eye, and it would just go straight through into the blood and into the brain. And you would have these experiences, these hallucinogenic experiences. And after these people stopped dropping acid, they thought that they were clear of the effects of the drug, but they were wrong, because the lipid soluble myelin and the LSD would combine, and the LSD would stay resident in their brain for an extended period of time. And later in life, it could pop back out and produce flashbacks. Okay, so it could re-exert its effect. So you always want to be careful around lipid soluble compounds, because the body has an almost infinite capacity to store them. Okay. Another example of this, when I was in school many years ago, was if you couldn't afford to go to Florida on spring break, you would buy a product called Tan in a bottle. And this is basically large doses of vitamin A. Okay. And so you would order these capsules out of the back of a magazine, and they would come in a few days, and you would take these pills, and in a few days you would turn orange. All right. And then you'd go to school and say, look at my tan. But little known to you, you had actually taken a toxic dose of vitamin A. Okay. Vitamin A is one of the lipid soluble vitamins, A, D, E, and K. Okay. And the body can store it in toxic amounts. Right. You might be familiar with a medication called Retin A, which is used to control acne. All right. And one of the precautions that they tell you with Retin A is that you have to control your dose, because it can have effects on behavior and brain function. Okay. And that's because it's based on the lipid soluble skeleton of vitamin A. So the reason I bring this up is because we talk about pathogens, and there are some pathogens that are not living pathogens. Okay, they are agents of disease in the form of toxins, and you can lump lipid soluble material into that. Okay, So not to scare anybody, but a very popular fad right now is getting tattoos. All right. And tattoos use lipid soluble inks in some cases in order for the tattoo to stay in place. Right. If it was water soluble, it would just wash away. And I don't know what the effect of all those tattoos is going to be, but I will say that we're going to find out in a few decades whether or not that was a good idea. Okay. So lipid soluble compounds, you got to watch out. Okay. All right. Virulence and pathogenicity. Virulence is the degree of pathogenicity or disease producing power of a microbe. And that's basically determined by a virulence factor. Okay. Virulence factor. What is a virulence factor? It's a weapon. Okay. It's a weapon to produce disease. And these can be a variety of things. Okay. These can be toxins, these can be enzymes, this can be a lot of stuff. It could be the ability to live inside white blood cells, for instance. Okay. Pathogenicity is related to the number of microbes, the portal of entry, host defense, and intrinsic characteristics of the organism and the virulence factors, right? After the pathogens have gained entry into the body, they have to undergo adhesion and many other steps to be able to multiply and cause disease. Okay, so these virulence factors are what help them do that. Okay, adhesion is the ability to attach, right? You can't do anything to the host unless you latch onto it. If you can't latch onto them, you're just going to wash off, right? So it's the most critical step in an infection. Um, it can be removed by, if you don't attach, by ciliary motion, by urine flow, by the action of mucus, right? Bacteria have to bind to the host using things like pillae or fimbriae or membrane receptor sites. Worms do it by barbs and suckers, okay? And protozoa can do it by literally burrowing or chewing their way through tissue, all right, or by hiding inside cells. Adhesion can be nonspecific or specific, okay? Viral adhesion happens by engagement of the capsid with the host cell, often through the spikes, right, that engage the receptor and then the cell, the targeted cell pulls it inside. Non-specific adhesion involves things like hydrophobic interaction. There we go with lipid soluble interactions again. Electrostatic attraction, molecular vibrations, Brownian movement, and recruitment and trapping by biofilm. These are used by microbes to gain access to our body, right? Where specific adhesion is a lock and key relationship between complementary molecules on the pathogen and on the host, right? Usually the attachment becomes irreversible, okay? Examples of specific attachments, one example that jumps out is the ability of the AIDS virus to latch onto CD4 and helper T cells and get inside and turn the helper T cell into a virus factor, right? Colonization is just the growth on or in the host, right? Human pathogens colonize tissues that are in contact with the outside world. We talked about what those are, right? Those are the ones lined with cutaneous or mucous membrane, they're listed here. Once you start to grow by getting in through the correct portal of entry and latching on, your growth is part of what can produce disease, right? Some virulent bacteria can produce special proteins that allow them to colonize these regions of the body and produce disease. An example, okay, would be something like Staph aureus, all right, which can grow on your skin alongside your good bacteria. And it can produce enzymes that digest connective tissue, like hyaluronidase, right? And when it does that, it chews through those protective layers and it could gain access to the blood supply and produce a blood infection, septicemia. Invasion, all right, can be aided by the production of extracellular material that disrupts the host cell membrane and breaks down primary and secondary barriers. and these are called invasins, right? This facilitates the growth and the spread of the pathogen as well. How do these pathogens escape your host defenses? Well, they got a lot of tricks, okay? They're sneaky. They can avoid contact with the white blood cells. They can inhibit the white blood cell's action. They can kill the white blood cell, okay? Or they can live inside it. They can produce products that damage the white blood cell while it grows inside it, or damage it before it gets a chance to eat it, okay? The ability to avoid the host response can be due to the presence of things like a capsule, a protective layer outside the microbe that makes it difficult to eat and destroy, right? It can produce proteins that bind to host cell antibodies, or it can mutate in order to change its surface molecules and, as a result, temporarily evade the immune system. We have to appreciate the fact that this is a strategy that pathogens have used for many decades to avoid host defense by changing their appearance, okay, which they sometimes do naturally in the course of infection, or sometimes as a result of mutations, which occur anytime there's cellular reproduction or there's a reproductive cycle like in viruses, okay? Various strategies are used by microbes to avoid being eliminated by the host phagocytes, but usually they involve blocking some of these steps of phagocytosis, right? And then we have toxins, right? And a toxin is a molecule that damages tissue, and it can either be part of the structure of the pathogen, or it can be something that gets secreted, okay? Organisms that make toxins are toxigenic. The underlying mechanism that produces disease is the toxin. Bacterial toxins can include lipopolysaccharide, which are part of gram-negative cell walls, or proteins that are released from the bacterial cell, okay? Another virulence factor is determined by the ability of the organism to make enzymes and exotoxins that can damage host cells and interfere with vital cell functions. Toxins are a major virulence factor, okay? Let's give an example. We could look, for instance, at diphtheria toxin, okay? Diphtheria toxin is an exotoxin, and when it acts on the cell, it kills it, okay? Exotoxins are proteins that are released by bacteria during their exponential growth phase, and they act at sites other than the location of the infection. Toxins are often species specific and associated with a particular disease, okay? Clostridium tetani is the only organism to produce the tetanus toxin, and Corynebacterium diphtheria makes diphtheria toxin. As with other proteins, these enzymes can be denatured by heat, acid, and proteolytic enzymes, which break down the primary structure of the protein. Like enzymes, a lot of these exotoxins are substrate specific, and generally, the site of damage caused by the toxin indicates the location of the substrate for that particular toxin. This has led to naming these toxins according to their target, such as enterotoxins, which target the GI, neurotoxins that target nerve tissue, leukocidins, which destroy white blood cells, or hemolysins, which rupture red blood cells. Okay? An endotoxin is part of the structure of the bacteria itself. Okay, an example would be lipopolysaccharide in the gram-negative cell wall that gets released when the bacteria lyses, when it blows up. Okay, it can act on sites remote from the original infection, and they tend to be less potent and less specific than an exotoxin, right? Because these toxins are not proteins, they're heat stable, and even boiling for half an hour doesn't destabilize them. However, certain oxidizing agents like superoxide, peroxide, and hypochlorite have been reported to be effective in neutralizing them. The figure below shows the action of exo and endotoxin. Exotoxins on the left are made within the bacterial cell and released into the environment, while endotoxins are only produced when the bacterial cell actually ruptures. Okay? The portal of exit is usually the portal of entry, but it can be other portals as well. Portal of exit is just how you get out of the host. Pathogen can leave by excretion and secretion as well. The excretory portals of exit are defecation or through the blood. The secretory portals are nasal secretions, saliva, sputum, respiratory droplets, tears, and earwax. And these are also ways by which pathogens can exit a host and infect another, usually by casual contact. Okay. All right. Getting close here. So when we come back from break, we're going to talk about Koch's postulates. We're going to talk about epidemiology and reservoirs. Okay. We're going to talk about the different ways that disease transmission can take place. Okay. And I'll also go through and correct that last quiz you took so that the grades reflect the questions you actually answered. Okay. So let's break for 20 and then we'll come back. Disease and the patterns of infection. Not all infections cause disease, and it's essential to determine the difference between an infection and a disease. An infection is the colonization of the host by an agent. When an infectious agent causes pathological change and interferes with normal functioning of the body, is termed an infectious disease. The study of the cause of disease is called etiology. Patterns of infection include local infections, where the organism enters the body and remains confined to a tissue, focal infections, where the pathogen spreads from a local infection to other tissue, and systemic infections, where it spreads to many sites and tissue fluids, usually using the circulatory system. Septicemia is an example of a classic systemic infection because you've got pathogens growing and thriving in the blood, and they're going to spread to every organ in the body, and that can result in multiple organ failure and death, right? Bacteremia is just the presence of bacteria in the blood. Toxemia, the presence of toxins in the blood, and viremia, the presence of viruses in the blood. In case you haven't figured out, the suffix emia means in the blood, okay? So just keep that in mind when you see it. Let's circle back. In blood. They're not going to like it because it won't fail them all. Okay. So some patterns of infection include mixed infection, where you've got several infectious agents concurrently establishing themselves at the same site. Acute infections, which come on rapidly with severe symptoms and vanish. Chronic infection, which are usually less severe symptoms, but they persist for a long time. A primary infection, which is an initial infection, and a secondary infection that follows the primary. Okay, now this is an important aspect here. A lot of times you'll get like a viral infection, and the doctor may give you an antibiotic. Right now, an antibiotic is to act against bacteria and doesn't do squat for viruses. So why is he giving you an antibiotic for a viral infection? And the answer is because he wants to reduce the likelihood that a secondary infection will set up. A secondary infection is just going to be the bacteria coming in to vulnerable tissue that's been damaged already by what the virus has done. Okay. A subclinical infection doesn't cause any apparent symptoms and can be carried for a long time. Okay. In mixed infections, sometimes you get synergistic action where they damage tissue more effectively as a team than they do on their own. In a primary infection, the initial infection can be followed by the complications due to another microbe. In general, secondaries are caused by different microbes, which gain access to the body through a lesion of either the skin or the mucous membrane damaged by the first one, or they can be microbes of the normal flora that become opportunistic pathogens. Okay. Now, Koch's postulates were developed way back around the time that the the modern microscope was being developed. Okay. And this was the idea that we could figure out what's causing the disease by applying these particular postulates, right? The suspected pathogen has to be present in every case of the disease. The pathogen has to be isolated from the diseased host and grown in pure culture. The pathogen from pure culture has to cause the disease when inoculated into a healthy experimental host. And then you got to re-isolate the same pathogen from that freshly infected host. So none of this was possible before the microscope because we couldn't see what we were looking for, right? But now, at this time, once the microscope had been developed, we could actually observe the microbe that could be responsible for producing the infection, okay? This was proposed as a series of postulates by Pasteur and Koch. Although Pasteur was convinced that microbes cause disease in humans, he wasn't able to link a microbe to a particular disease. And after many years of experimentation, Koch was able to establish that an organism can cause a specific disease and develop these postulates to determine this. Using his postulates, he proved that Bacillus anthracis is the causative agent of anthrax, and Mycobacterium tuberculosis causes TB. This concept is now known as Koch's postulate. So you can see here, again, how we're operating, right? You've got the suspected pathogen. You isolate material from the disease, the initially diseased organism. You look at it under the scope. You see what you can find. You grow it up in the lab. You infect a healthy host. You produce the same signs and symptoms. And then in every instance of the disease, you re-isolate the same organism. And that tells you what you got, right? But there are exceptions, right? Some pathogens can't be grown in the lab. Many infectious diseases can be caused by different pathogens. A particular pathogen can cause different diseases depending on how it gets into the host, and some diseases are caused by a combination of pathogens, while some pathogens are only going to act on humans, so we can't do Koch's postulates. It's ethically prohibited, okay? There's no animal host to substitute for a human, okay? Although Koch's postulates don't always seem to apply in these cases, scientists have found other means to reach the desired outcome by following other routes. Koch's postulates work in the food industry where microbes need to be carefully monitored and the desired microbe has to be isolated in the desired end product. Despite challenges, the postulates remain useful for confirming microbial roles in infection, disease, and other processes. Okay. So let's just take a look at epidemiology as an overview. Okay. Regulators make decisions on what they're not allowed to do, like smoking in public places or driving gasoline air-polluting cars. Hold on just a second. Let's deal with the sound here. That's better. Come on. There we go. Yeah. Yeah. Now we're ready. Oops, hold on. Get out of here. Sure. this is better.
How do regulators make decisions on whatever not, like smoking in public places or driving gas, dust, and air polluting cars? And how do health organizations decide what healthy eating recommendations to make, like having 5 servings of fruits and vegetables a day? These are examples of public health decisions based on epidemiology, the science of understanding how what we're exposed to or what we do may affect the overall health of society. But to make sense of epidemiology, we need to dig a little deeper. Let's start by going back to basics and ask, how do we work out whether something we do or are exposed to is harmful, and how harmful is it? The only reliable way is to use science to develop evidence that shows something is bad for health. But this isn't as easy as it sounds. One way is to do research on animals, called in vivo studies, or on cells in test tubes and petri dishes. These are called in vitro studies. But such research can only tell us so much. For example, many substances can affect us differently than they affect animals. So while chocolate is harmful to dogs and aspirin is toxic to cats, both are safe for humans when used appropriately. As for in vitro studies, cells that are isolated in the lab behave differently than cells inside the body. So we have to be very careful applying the results of such studies directly to human health. Of course, the most straightforward approach is to do experiments on real people, but with a few exceptions, this is highly unethical. We can't go around exposing people to potentially harmful substances just to see what happens. This leaves us with observing the real world, measuring what a lot of people are exposed to, and by a lot we mean tens and hundreds of thousands of people, and trying to work out what the connections are between these exposures and their health. Studies like this form a big part of epidemiology, and it is a powerful way to understand how exposures and behaviors potentially affect large groups of people. But it's also sometimes difficult to make sense of. To start with, just because someone was exposed to something and they got sick doesn't mean the two events are related. The exposure may not have caused the sickness. For example, lots of people eat ice cream when it's hot, and lots of people get sunburn when it's hot. But ice cream clearly does not cause sunburn. These events are instead correlated, meaning they often happen together. But they're not causative, meaning that eating ice cream doesn't directly cause sunburn. And while this example is pretty obvious, making sense of epidemiology data is often really hard, and care needs to be taken that we don't jump to the wrong conclusions. When examining relationships between exposures and health outcomes, there is a number of reasons why we might see an association. These include an actual cause, pure chance, bias, and what epidemiologists call confounding, other things interfering with what we observe. Bias can come about because of errors in how a study is designed. For example, if we observe only people who eat a lot of ice cream and live in really hot, sunny areas, and don't include anyone else that doesn't eat so much or live elsewhere, the association between ice cream eating and getting sunburn will seem very strong. In other words, the results will be misleading. They'll be biased toward a particular, and in this case, wrong conclusion. Confounding, on the other hand, is where other factors confuse our interpretation of exposure and outcome. For example, imagine a study that suggests people who drink more coffee are more likely to develop heart disease. It'd be tempting to conclude that coffee causes heart disease, but people who drink coffee also tend to smoke. In this case, smoking is a confounder. Since it is associated with both drinking coffee and heart disease, it can make it seem that coffee causes the condition if we don't take smoking into account. In real life, there are many confounders, some more obvious than others. Because of this, epidemiological investigations take quite a bit of detective work to figure out what exposures really lead to the health outcomes and which ones only appear to. To tease out what is relevant and what is not, epidemiologists use statistics. One standard practice in analyzing data is to look at the probability or p-value to determine if the findings are likely to be true or are simply due to chance. The lower the p-value, the more likely it is that the results of the study represent reality and did not just happen because of chance or random variations. Usually, epidemiologists consider a p-value of 0.05 or lower as indicating that the study results are statistically significant, which is just a fancy way of saying that there's less than 5% chance of these results being due to random variations. However, the p-value only helps you get a sense of whether study outcomes are due to chance or not. It does not help us examine how strong the association is or how important the health implications are. And if statistics aren't done well, even low p-values can be misleading. Consider cancer risk. Epidemiological work shows that both smoking and processed and red meats have a statistically significant association with increased cancer risk, meaning they both have p-values below 0.05. But while smoking increases your chances of getting cancer by around 20 times or 2,000%, eating red and processed meats increases it by only 20% or 0.2 times. This is extremely low when you consider all the other things you're exposed to that potentially impact your health, especially if the chances of getting cancer aren't high to start with. And this is why looking at the p-value on its own is not enough. And researchers also need to consider how large of an effect an exposure has, called effect size, not just whether there's likely to be an effect or not. Lastly, when making sense of epidemiological studies, it's important to remember that this science deals with the collective health of thousands and millions of people and not individuals. Because we're all different and live under different conditions, it is very hard to apply broad conclusions from such studies to single people. But they are good at indicating what whole communities should do to stay healthy. And just to complicate things further, epidemiology studies may not include people like you, meaning that they may be less important to you than to others. The bottom line is that it takes a lot of work to conduct epidemiology studies well, and it takes a lot of work to interpret them correctly. Despite these difficulties, epidemiology is crucial for making public health decisions and improving the well-being of people, so that on balance, we all live healthier lives. These decisions do not only mean better healthy lifestyle recommendations and programs, they're also important for reducing healthcare bills and increasing productivity over tens of millions of people. Which is why, even though it's complicated, epidemiology is so important.
So this is the study of the spread of disease in populations. It's of major importance to state and federal public health departments. The CDC is the central source of epidemiological information in the US. Morbidity and Mortality Weekly Report contains data on morbidity, the incidence of specific notifiable diseases, and mortality. A well-developed network of individuals and agencies keeps track of infectious disease at the local, district, state, and national and international level. The CDC shares its information and statistics with the WHO for the purpose of worldwide tabulation and control. It's the study of the distribution of the cause of disease in populations, serving as a foundation of logic of intervention needed in the interest of public health and preventative medicine, how many people are affected, where the outcome of the disease is going to end up. You're going to recover, you're going to die, you're going to have a disability. Okay. They use three basic approaches to study the dynamics of disease in populations: descriptive epidemiology, an analytical epidemiology, and experimental. Okay. With descriptive, we collect the data about the disease, including info on the affected individual, the location, the period of disease occurrence, the age, the gender, the occupation, the health history, sexual behavior, eating habits, and socioeconomic status of those affected, and the time course and the chain of transmission, including what's known as the index case, which is the first reported case. Now, it may not be the first case of the disease, but it's the first case that's notified, right? Seasonal variations in climate can be a consideration. The time course and the chain of transmission are important in descriptive epidemiology, and investigators make every effort to ID the index case which is the first reported case of the disease. This can be difficult because the victim of the disease may have recovered, moved, or died. Okay. Analytical epidemiology attempts to demonstrate a cause and effect relationship using data collected in descriptive epidemiology. It's retrospective, the data analyzed are after the fact, and prospective, the data are collected during the epidemic. So you do both. Okay. Analytical epidemiology is often applied when it's unethical to apply Koch's postulates because the disease is a human disease only and doesn't have an animal model. Experimental epidemiology starts with a hypothesis about a disease and is followed by the design of experiments and studies to test it, right? The application of Koch's postulates is how we do this, okay? Diseases can be reported by the prevalence of the disease, right? Which is the total number of cases in the whole population, or by the incidence of a disease, which is the number of new cases over a certain period of time compared with the general healthy population. Okay, so you can see the formulas below, right? Prevalence, total number of cases over a total number of persons times 100, that's the percentage. And the incidence, the number of new cases over the total number of susceptible people, okay? Changes in the prevalence and incidence of disease are generally monitored every season, annually, and long-term to predict a trend. These monitoring stats make it possible to define the frequency of a disease in a population. The frequency and geographical location and the occurrence of a disease allow for classification of the disease as an endemic, a sporadic, an epidemic, or a pandemic disease. Okay? An endemic disease is repeatedly present in a given population or geographic area. A sporadic breaks out occasionally, and an epidemic occurs with increased frequency above what's usually experienced in a population of a given area, while a pandemic is worldwide. An example of an endemic disease would be the common cold. A sporadic disease would be typhoid. Flu is an example of a disease that can occasionally reach epidemic proportions. And of course, COVID is a pandemic, okay? Reservoirs are just areas where the pathogen hangs out, all right? For an infection to continue, the causative agent has to have a permanent place to exist. Sites where pathogens are maintained and are a source of infection are called reservoirs. Okay, these can be animal, human, or non-living. Animal reservoirs, diseases that occur primarily in animals, can be transmitted to humans, and they're known as zoonoses. Okay, the roots include direct contact with the infected animal, contact with the animal waste, contact contaminated food or water, dust from contaminated hides, fur, or feathers, and by consuming infected animal products or by insect vector. Okay, human carriers are the main reservoir for human infections. Infected individuals that don't have symptoms are known as asymptomatic carriers, and they are the most dangerous individuals in an instance of a disease because they're not aware that they're sick and they can spread to healthy individuals. Okay, we're going to see a shocking example of an asymptomatic carrier that is colloquially known as Typhoid Mary here in a bit. So keep that in mind. Non-living reservoirs, soil, water, food. Soil is a great environment for pathogens like fungi, helminths, and bacteria. Okay. People with obvious infectious diseases are easily identified and precautions can be put into place to avoid transmission, but an asymptomatic carrier is like a silent agent, like a silent terror cell, okay, and can spread the disease without even them knowing it, okay. It's believed that these healthy carriers have immune system responses that prevent them from getting the disease. If the soil is contaminated with fecal material, and if you're looking here at non-living reservoirs, it can contain organisms normally present in the GI tract of humans and animals, and ultimately is responsible for GI disease. In addition, the soil can harbor pathogens like Clostridium botulinum, which causes botulism, and Clostridium tetani, which causes tetanus. Water can potentially be contaminated with fecal material and urine containing parasitic worms, eggs, pathologic protozoan, bacteria, and virus. Contamination with infectious agents can also include foods improperly prepped or stored. Okay, so an example, right? Diseases spread by what we would call fecal oral route. Okay, one might be salmonella. Okay, as an example. Infectious diseases can be spread by a reservoir or portal of entry to a portal of entry of another host, or you can do auto inoculation, where you actually introduce the organism by touching a part of your body that lets it inside from a part of your body that has the pathogen on it, right? Infectious agents can be spread by contact, vehicle, or vector transmission. Contact transmission, there's a variety of ways, right? In direct contact, you come into physical contact with the pathogen between hosts without an intermediate. Person to person can happen by touching, kissing, or intercourse. Examples: respiratory tract infections can be spread by aerosols, staphylococcal infections by infected fluids, right? Measles, scarlet fever, and STDs. In direct contact, the pathogen is transmitted by a fomite, which is a non-living object, doorknobs, tissues, handkerchiefs, eating utensils, and so on. And in droplet transmission is transmitted through the respiratory tract by droplets. Okay, one of the most amazing things is that if you sneeze and you don't cover when you sneeze, the droplets can be suspended in the air for up to 20 minutes. Okay. When we look at contact transmission, okay, zoonoses are an example. They can be transmitted from an animal reservoir to a human by direct contact through touching, biting, or scratching. Okay. Direct transmission is possible within a single individual. That's auto inoculation. Okay. And then indirect and droplet. Okay. Vehicle transmission requires an intermediate. Okay. In airborne, by droplet nuclei, you inhale it or ingest it. Okay. Through dust, by sweeping, mopping, or changing bed linens. You have to travel over a meter to be considered an airborne transmitter. Okay. Waterborne, through untreated or poorly treated sewage. GI diseases like giardiasis, amoebic dysentery, and cholera are through fecal oral route. Waterborne shigellosis and leptospirosis are examples of waterborne. Pathogens shed in fecal material can enter the GI tract mucosa or the skin and cause disease in other areas. Modes of transmission in this category include bodily fluids and IV fluids, foodborne, waterborne, airborne, right? Pathogens such as schistosoma and enterovirus are shed in feces and get into water supplies and enter the GI mucosa of the skin and then cause disease elsewhere in the body. Okay. Foodborne, okay, can come from completely cooked food, poorly processed food, or foods that are prepped under unsanitary conditions, okay, or not refrigerated or poorly refrigerated. Those are all possibilities, right? Bodily fluids, especially among healthcare workers, which is why we need to take universal precautions when handling fluids that potentially are contaminated with pathogen, including things like blood, urine, saliva, and other bodily fluids. Okay. Examples, right? Viruses such as Hep A can be transmitted by vehicle. Diseases that can be transmitted also include HIV, Hep, and herpes through bodily fluids. Right? Vector transmission is where we have an intermediate, usually an arthropod, an insect that spreads it. Okay. Mosquitoes frequently spread these types of diseases. Ticks, okay. Sand flies. Animals, especially arthropods, can be biological vectors. Okay. They transmit the pathogen, but also serve as a host for part of the pathogen's life cycle. Biting insects, including mosquitoes, ticks, lice, fleas, and blood-sucking flies, or bugs and mites are examples. while a mechanical vector is an organism that spreads it by contact, right? So it lands on a contaminated surface and then lands on you or lands on your food. An example would be something like a housefly. Okay. Nosocomial infections are acquired in a healthcare setting. That's why they're called nosocomial, right? They're secondary to the patient's original condition. Nosocomials are among the top 10 leading causes of death in the US. The CDC developed the National Nosocomial Infection Surveillance System in the early 70s to monitor incidents of nosocomial infections and their associated risk factors and their pathogens. The purpose is to help infection control professionals and hospitals stay up to date on rapidly expanding science and practice of infection prevention and control, and to be better able to manage endemic and epidemic episodes of nosocomial infections. What are some of the different types, right? Healthcare facilities are filled with sick people capable of shedding a variety of pathogens from different portals of exit, okay? So this can contribute. People have stress, weakened, or compromised or suppressed immune systems, okay? So that can promote the growth of opportunistics. And they're more susceptible to pathogens and opportunistics, all right? Exogenous nosocomials can be caused by pathogens in the healthcare environment that are shed by sick people, while endogenous nosocomials are caused by microbes in the normal flora of the patient. They can become pathogenic for a variety of reasons. You also see immune system decline in these patients, often due to stress. And the use of antimicrobial drugs can select for resistant versions of microbes that can produce nosocomials. You can also get overgrowth and superinfection as a result of this. Now, an example, right, of this is something called MRSA, okay? So if we look here, not plugged in. MRSA, MRSA, okay, is an example of a nosocomial infection that was produced by natural selection through the use of methicillin. The acronym stands for methicillin-resistant Staph aureus. And it's frequently a problem in surgical site infections and UTIs and in a variety of other nosocomials. And it's so prevalent now that insurance doesn't even cover it, okay? It's the job of the infection control officer to deal with things like that, okay? Super infections, all right, are happen in the absence of the normal competition of your normal flora, okay? An example, you might take a broad spectrum antibiotic to treat a particular disease and wipe out your normal gut bacteria, and then you could acquire C. diff, all right, subsequently. That being an example of a super infection. And it's not limited to healthcare settings, right? Iatrogenic nosocomials result from the use of procedures such as catheters, invasive procedures, surgery, okay? All of these are examples of accessing parts of your body where normally a microbe can't get, okay? They can result from medical procedures that expose sterile parts of the body, such as open heart surgery, right? Or it can be an indwelling device, okay, such as a catheter or an insulin pump or a port that you might use, for instance, in dialysis. Principal routes: direct contact transmission from hospital staff to patient, visitors and patients, patient to patient through activities of staff, indirect contact transmission through fomite, airborne transmission through ventilation, okay? Because of the large number of pathogens in the healthcare environment and the compromised state of the patient, the route and the chain of transmission constitute an ongoing challenge for healthcare professionals and facilities. Some hospitals are reserved for specialized care, including burn, hemodialysis, recovery, ICU, and oncology. Unfortunately, those also provide a risk for epidemic spread of nosocomials from patient to patient. Okay, so you got a vulnerable population, and you've also got resident microbes. Okay. Drug-resistant pathogens are becoming an increasing threat. Over 70% of bacteria causing these infections are resistant to at least one antimicrobial. Persons infected with drug-resistant organisms generally have longer hospital stays and require second or third choice drugs that can be less effective and more toxic to the patient. Okay, so this is a major problem. So what we want to
Or anyone in the healthcare field. To minimize the risk of infection, it is important that everyone follows standard precautions. To understand what standard precautions really means, let's take a look at how the Centers for Disease Control and Prevention define standard precautions. Standard precautions represents a system of barrier precautions to be used by all personnel for contact with blood, body fluids, secretions, excretions, non-intact skin, and mucous membranes, regardless of the person's diagnosis. This system includes the concepts of body substance isolation, which reduces the risk of transmitting pathogens from moist body substances, and universal precautions, which reduces the risk of transmitting bloodborne pathogens. There is a rising concern about exposure to blood due to bloodborne pathogens. You're probably asking yourself, what is a pathogen? Pathogens are disease-causing microorganisms that are found everywhere in the environment, on humans and animals. Pathogens found in the blood are called bloodborne pathogens. Some examples of bloodborne diseases are hepatitis B, hepatitis C, HIV, and malaria. Standard precautions is an approach to infection control that treats all human blood and certain body fluids as if they are infectious. Since these pathogens are too small to be seen by the naked eye, and you can't tell by looking at an individual whether they have a disease, you should protect yourself with simple and effective barriers. This is the rationale of standard precautions. Transmission-based precautions, in addition to standard precautions, are used when someone is suspected of being infected with a highly contagious pathogen. Transmission-based precautions are required where airborne, droplet, and contact transmission of infectious organisms may occur. Transmission-based precautions would be used if a person has chickenpox, measles, or an infection, to name a few. It is not always possible to know whether or not a person is infected with an infectious disease. Universal precautions is made up of a system of safeguards designed to help protect you and the people you support. Safeguards are things like personal protective equipment, safe work practices, good hand washing, good housekeeping, and vaccinations. All of these systems should be used together to form a protective barrier. We'll discuss these safeguards in more detail later. There are some people who may have an infectious disease and not show any symptoms or even be aware of their infection. For example, how many of you know the story of Mary Malone, also known as Typhoid Mary? Mary was the first person found to be a healthy carrier of typhoid fever in the United States. Typhoid fever is a life-threatening illness caused by the bacterium Salmonella typhi. You can get typhoid fever if you eat food or drink beverages that have been handled by a person who is shedding Salmonella typhi. Mary earned her living as a cook for private families. When several outbreaks of typhoid fever developed in homes where she worked, the public health officials questioned her. Under questioning, she stated she rarely washed her hands when cooking and felt it wasn't needed. She also insisted she never had typhoid fever and had always been healthy. However, the health officials linked the outbreaks back to her. After her death in 1938, the autopsy revealed that her gallbladder was still actively shedding typhoid bacteria. This example helps us to understand the importance of following standard precaution procedures at all times. Somehow, pathogens need to get inside your body to cause an infection. Let's take a few minutes to discuss how these pathogens can get inside your body. It can happen from a sharp object contaminated with the infectious material, such as a needle, broken glass, wires, or any other object that can break or cut your skin. From openings in your skin, this would include cuts, nicks, abrasions, skin infections, acne, and surgical incisions. Through your mucous membranes. Mucous membranes are the moist surfaces of your mouth, eyes, and nose. By touching contaminated environmental surfaces and transferring the contaminant to mucous membranes or open skin, surfaces can be heavily contaminated without visible signs of contamination. Diseases that are transmitted from other people or animals are called communicable diseases. Some examples of communicable diseases are measles, strep throat, and colds. Standard precautions should be used in the care of all people and used regardless of whether or not the person is known to have a communicable disease. In order for a disease to spread to others, certain conditions are needed. This is sometimes called the chain of infection. Let's take a few minutes to discuss these conditions. First of all, there must be an agent like a virus, fungus, or bacteria that can cause the illness. The agent also must have a place to live. This is called a host. Humans, animals, and insects are the most common hosts, although agents can also live in the environment. The agent must also have the right environment to survive. Often moist, dark places help them to live and grow. The agent must be able to enter the host. For example, a person who has a break in their skin or someone who breathes in the agent. An agent must have a way to move from its host to infect others. This is called transmission. Some examples are through blood, body fluids, a sneeze, or a cough. There also must be a person who can catch the disease. If someone can't fight off the germ, they can become infected. This is called being susceptible. This completes a chain of infection. The cycle continues as the agent is passed on to another host. The easiest way to remember how diseases can spread is through the David method. Communicable diseases are mainly spread in the following ways. Direct. This is through person-to-person contact. Airborne. This simply means through the air. These infectious droplets remain suspended in the air because they are carried by microscopic droplet nuclei or dust particles. They can land on or be inhaled by the host. Vector. This occurs when an organism, such as a tick, carries disease-causing microorganisms from one host to another. Indirect. This occurs when microorganisms are transmitted by contaminated items such as food, water, medication, and equipment. In other words, person, object, person. Droplets. When someone coughs, sneezes, or talks, tiny droplets may be generated. These infectious droplets propel a short distance through the air and land in the host's eyes, nose, or mouth. Droplets do not remain suspended in the air. The spread of infectious diseases can be stopped by killing the agent, changing the environment so the agent can't survive, wearing protective gear, removing the agent's means of spreading, and making sure people are immune to the agent through immunization. This is commonly called breaking the chain of infection. The most important and effective thing you can do to help prevent the spread of infection is to wash your hands. Hand washing is often taken for granted because it can be routine, but good hand washing stops the spread of contaminants from your hands to other parts of your body or other surfaces you may come in contact with. Let's take a few minutes to go over some general guidelines to follow when washing your hands. If you remove your gloves, wash your hands thoroughly. If skin comes into direct contact with blood or other infectious material, wash with soap and water as soon as possible. If you are using antiseptic towelettes or hand sanitizers, you should still wash your hands as soon as possible after touching something that is visibly contaminated. Remember, good hand washing doesn't take a lot of time. Follow these 6 steps for good hand washing. Remove any jewelry you are wearing if possible before washing your hands. Bacteria can thrive under rings and watches. When washing your hands, use warm, running water. Wet your hands and apply soap. Use plenty of soap to create a good lather. Vigorously rub together all surfaces of your hands for 15 to 20 seconds. Friction helps remove dirt and microorganisms. Pay close attention to cuticles and under your fingernails. Keep splashes to a minimum and try not to touch the sink itself. Rinse your hands, keeping your hands lower than your elbows. Dry your wrists and hands completely with a clean paper towel. It is best to pat your hands dry rather than rubbing them. This helps prevent your hands from chapping and cracking. Turn off the faucet. Remember, don't touch the faucet with your clean hands. Use a clean paper towel. Remember these 6 steps the next time you wash your hands or assist the people you support to wash their hands. You're probably now asking yourself, how often should I wash my hands? The following are general guidelines for washing your hands. When you arrive at work, before and after assisting someone with personal care, before and after wearing gloves, before and after you eat, before and after cooking, before leaving work, after blowing your nose, touching your face or hair, after using the bathroom, after any cleanup you do, after smoking, or whenever your hands are soiled. Standard precautions requires the wearing of personal protective equipment whenever you anticipate the possibility of splashing or spraying of blood or other body fluids, such as mucus, drainage, or emesis. Personal protective equipment helps protect you from contact with blood or other potentially infectious material. Personal protective equipment includes gloves, masks, gowns, aprons, face shields, protective eyewear, mouthpieces, and resuscitation bags or other ventilation devices. Personal protective equipment must be appropriate for the task. You should wear as much as you anticipate you will need to keep blood and other infectious materials off your skin, mucous membranes, or clothing. Anytime the personal protective equipment is torn or contaminated, remove the equipment and replace as soon as possible. Gloves should always be used whenever you anticipate contact with blood, potentially infectious material, mucous membranes, or non-intact skin. When working directly with people, single-use disposable gloves should be worn. Single-use disposable gloves are just that. They should be used only once and then disposed of immediately. The wearing of gloves does not substitute for good hand washing. It's not acceptable to wear the same gloves or wash gloves between each person. Another type of glove you may use is a utility glove. Utility gloves are generally used during housekeeping. Utility gloves should be cleaned after every use with soap and water and then dipped in bleach solution. The gloves should be worn, not handled, during this cleaning and sanitizing procedure. Utility gloves are not used when working with people. They are only appropriate for cleaning. Because gloves can be torn or damaged, always bandage any cuts or open areas on your hands before applying gloves. Replace the gloves as soon as possible if they are contaminated, torn, punctured, or damaged in any way. Follow these general guidelines to determine when to wear gloves. Anytime you are handling blood or body fluids. Anytime you have open wounds on your hands. Anytime there's potential to come into contact with objects contaminated with blood or body fluids. If you are touching mucous membranes. When touching non-intact skin of any person. Anytime you are cleaning contaminated or potentially contaminated surfaces, equipment, or objects. Good housekeeping helps protect everyone and is everyone's responsibility. Carefully follow your agency's policies and procedures regarding handling infectious materials. The following are some general rules when dealing with infectious materials. Always wear gloves when handling infectious materials. Handle used equipment soiled with blood or other potentially infectious materials with care. Do not let the equipment touch your skin, mucous membranes, clothing, or other items in the environment. Do not eat, drink, smoke, apply cosmetics or lip balms, or handle contact lenses where there is reasonable chance you have been exposed to blood or body fluids. Do not keep food and drink in places where blood or other potentially infectious materials are present. Clean reusable equipment appropriately before using it for another person. Single-use items should be discarded. Be careful to prevent injuries from needles and other sharp instruments when using, cleaning, or disposing of them. Never bend, recap, or break needles after use. Dispose of contaminated sharps immediately after use in appropriately labeled or color-coded, leak-proof, and puncture-resistant containers. Report any container that is full. Remember, safety razors and razor blades are also considered sharp material. Follow your agency's guidelines when disposing of sharp materials. To prevent contamination, handle soiled linen as little as possible and with minimum agitation. Gloves should be worn. Without sorting or rinsing, place soiled linens in a leak-proof bag in the area where it was used. Follow your agency's guidelines for handling contaminated linens. Transport specimens in closed containers. Wear gloves and handle the containers carefully. Specimens taken to the doctor should be placed in a closable plastic bag. Carry waste containers away from your body. Clean all blood and body fluid spills promptly according to your agency's policy. Everyone should know what to do if an accidental exposure of blood or body fluids occur, such as a needle stick injury or a splash into the eyes or mouth. This is called a significant exposure. If you are involved with a significant exposure, don't panic. Immediately wash the affected skin with soap and water. Flush affected mucous membranes, such as eyes, with water. Report the incident to your supervisor immediately. There are a number of steps that must be taken in the first 24 hours to decrease your risk of becoming ill, so quick reporting is essential. Follow your agency's guidelines when dealing with any accidental exposure to blood or body fluids. If there is a blood spill, your agency may have a blood spill kit for your use. Follow your agency's policy for using your specific kit. Infection control practices are easy to ignore because they can be so basic, but you must take the time and make the effort to consistently follow infection control procedures. Let's take a few minutes to summarize some of the main points when following standard precaution guidelines. Pathogens are disease-causing microorganisms that can be found everywhere in the environment, on humans and animals. Standard precautions is an approach to infection control that treats blood and body fluids as if they are infectious. Standard precautions includes the concepts of universal precautions and body substance isolation. It is not always possible to know whether a person is infected with a disease. The person may not show any signs or symptoms or even be aware of their infection. Standard precautions are made up of a system of safeguards designed to help protect you and the people you support. These safeguards include personal protective equipment, safe work practices, good hand washing, good housekeeping, and vaccination. All must be used together to form an effective barrier. Pathogens need to get inside of your body to cause an infection. Some ways they may enter your body are by punctures from sharp objects or openings in your skin. Pathogens can also enter through mucous membranes of your mouth, eyes, nose, or by touching contaminated environmental surfaces. In order for a disease to spread to others, certain conditions are needed. These conditions include an agent, like a virus or bacteria that can cause illness, a host, a portal of entrance, a means of transmission, and a susceptible person. Transmission of pathogens can occur by direct, airborne, vector, indirect, or droplet means. The most important and effective thing you can do to help prevent the spread of infection is to wash your hands. Standard precautions require the wearing of personal protective equipment whenever you anticipate the possibility of splashing or spraying of blood or other body fluids. Following standard precaution guidelines is crucial for protecting you and the people you support from potential infection.
All right, let's take a 15-minute break and then we'll come back with Q&A.